Hello, everybody. I'm delighted to introduce Professor Helder Maiato, our first keynote speaker of this virtual conference on cell and developmental biology. Just a brief overview of Helder. Helder graduated in biochemistry and holds a PhD in biomedical sciences from the University of Porto. He was a visiting PhD student at the University of Edinburgh in the UK in the lab of Bill Earnshaw, and a postdoctoral research affiliate at the New York State Department of Health in the USA in the lab of Conley Ryder. In 2015, he was honored with the Young Investigator Award from the Louis Jeantet Foundation and was elected as an EMBO member in 2016. Currently, he is coordinating investigator at the Institute of Research and Innovation in Health and invited auxiliary professor with habilitation at the Faculty of Medicine at the University of Porto. His key research interest is the spatiotemporal control of chromosome segregation during cell division, and has been the coordinator of 2 projects from the European Research Council on this topic. Together with his wife, they founded Y-Science, a foundation that brings together scientists to promote science education and the scientific method in young children, which I know is close to both their hearts. So now I'll hand over to Helder, to present his work on the Tubulin Code.
Hello, good morning. My name is Helder Maiato. I work at the Institute for Research and Innovation in Health at the University of Porto. I'd like to thank the organizers for giving me this opportunity to present our latest work, and it's entitled the Mitotic Error Code. It's of course oriented for mitosis aficionados, but I hope to raise your interest as it touches many aspects of microtubule function regulation that I think might apply to other fields as well. So why do we study cell division? It's, of course, a fundamental process in our bodies. Cells need to divide to make multicellular organisms, and that's why we're here today. And so to give you an idea, about 1 trillion cell divisions take place in one's body every day. So that's a lot of cells dividing that we need to ensure very fundamental functions also in the human adult organism, such as the renewal of epithelia, operating our immune system, repairing tissues, etc. And if you take into account that so many cell divisions are taking place, and based on what we know in terms of potential chromosome missegregation events that happen during cell division, we can easily understand that if we have one potential missegregation event happening every 200 divisions, and these are based on in vitro studies, of course, if we do make the math and combine these 2 numbers, 1 trillion cell divisions every day, and one potential deleterious effect caused by the loss or gain of a particular chromosome, we can easily understand that our cells and we as organisms live under the constant threats of missegregating a chromosome. And what's the big deal about it? Well, losing or gaining chromosomes is just the most common feature in human cancers. It's the most horizontal property of human cancers. There's a lot of debate whether this can trigger cancer on its own, but it's pretty consensual now that gain or loss of chromosomes during cell division and giving rise to so many chromosome aberrations in human cancer is really an important feature for tumor evolution. It's a very fast way of evolving a cell, because all of a sudden you lose or gain or amplify several genes in a single step. So you can imagine that this is actually quite important to understand, and based on World Health Organization numbers, we estimate that about 10 million people every year die of cancer. And just to give you a comparison for these numbers, we live very different days, days with COVID-19, and it's just about 20 times more people dying from cancer than have so far died from COVID-19. So puts the bar pretty high in terms of social relevance for understanding chromosome loss and gain and what are the mechanisms to prevent these to happen. And this is critical because we all know that we not all die from cancer, despite the fact that we are so prone to lose or gain chromosomes. But mitosis is actually a very complex system, very dynamic. And here I'm illustrating a movie of a cell dividing where the mitotic spindle is shown in green, expressing a GFP fusion of microtubules, and the red dots that you see are the kinetochores. The kinetochores are the interface between the chromosomes and the spindle microtubules, and are absolutely essential to segregate chromosomes during mitosis. One can easily appreciate how orchestrated this process has to be when we transform these movies in 2 dimensions. So in the x-axis you see time, and in the y-axis you see length. And you can see that the different stages of mitosis here depicted in different colors. The chromosomes start by meeting all at once at the spindle midzone, establishing the metaphase plate, and this process is known as chromosome congression. And only when they congress and establish this position at the equator of the cell, they are allowed to separate their sister chromatids and migrate quite synchronously and uniformly towards opposite sides of the cell, giving rise to 2 daughter cells that are exactly or are supposedly genetically identical and with the exact same copy of chromosomes as the mother. So the topic of the talk that I will cover today starts with a story in which we try to understand this first stage of mitosis before the sister chromosomes actually separate. So this point of congression at the equator, and what are the mechanisms underlying this very fundamental mechanism. So in a very short summary, I'm giving you this motor-centric view of the problem. So there's motor proteins, namely minus N-directed motors of like dynein, and plus N-directed motors from the kinesin family, and one of them is SEMP-E, which is plus N-directed motor, which sits on the kinetochores and uses pre-existing microtubules to slide the chromosomes that are at the poles towards the equator, migrating the chromosomes to where they should be positioned before starting their anaphase movement towards opposite poles. Just before I forget, the dynein motor, it's also sitting at the kinetochores, but it has a different preference in terms of direction. Rather than moving cargo towards the plus ends of the microtubules, this motor prefers to move the cargo towards the minus end of the microtubules, so very close to where the mitotic spindle poles here, represented by the centrosome, would be positioned. Then there's another class of proteins, also from the kinesin family, that rather than sitting on the kinetochores, they actually sit on the chromosome arms. But they also have plus N-directed activity, and they assist chromosome congression by participating in what is known as this polar ejection pores. If you imagine the mitotic spindle pole as the north pole or south pole of our planet, so you see that if there's what we call polar winds, these winds will push the chromosome arms mediated by these motors and help assisting the congression of these chromosomes towards the equator. So you can see that there's 3 main motors involved in the process, 2 of them located in the exact same position at the kinetochores, but with completely opposite activities, and then a family of motors associated with the chromosome arms. So the question that arose at the time was that how were these opposite and spatial distant activities coordinated to drive chromosome congression during mitosis? So if we let congression depend on SEMP activity, and I'm showing you here a cell that has been treated with a SEMP inhibitor again, you can see that most chromosomes align and these chromosomes remain locked at the poles because SNP activity is inhibited. But then when we wash out the inhibitor, all these chromosomes at the pole, they always, 100% of the cases, they undergo motion towards the equator, and then the cell resumes division. However, if we now, for example, replace SNP motor at kinetochores by a conventional kinesin, so this classic kinesin 1, this is experiments by Michael Lamson's lab that we highlighted recently in the News and Views, where they basically compare how congression is if we let the process depend on a conventional kinesin compared to the SNP kinesins. And basically, the outcome was very interesting. If you let the system depend on a conventional kinesin, the chromosomes that are at the poles, then some of them, they do migrate towards the equator, but many of them are expelled towards the cortex. And as I showed you in this previous movie, if the system depends on SNP, the chromosomes that are at the poles, they always, once you engage SNP function, they always undergo directed motion towards the right, the correct side where the mitotic metaphase plate is positioned. So what's so special about SNP that appears to know where to go and guide and drive these chromosomes towards the equator? Just to highlight one aspect, because I also mentioned that these chromokinesins working on chromosome arms are also present and participate in chromosome congression. So we did one experiment to separate these functions. And it was a neat experiment because we basically use a laser microsurgery to separate the chromosome arm, which holds chromokinesins, from the kinetochore containing region that holds SNP. So these two movies that I'm going to show you highlight these procedures. So here's the chromosome arm pointed out by this arrow, and I'm going to sever this arm with a laser beam. And you'll see that when we do that, there's a little fragment here that was loose and immediately underwent congression towards the metaphase plate. So these chromosomes these cells have been treated with the SNP inhibitor. So you have these chromosomes at the poles that remain locked. So SNP is shut off, and that's why these chromosomes cannot undergo congression. But you notice that as soon as I release the chromosome arm from the locked motor, the chromosome arm was able to undergo directed motion, and in this case, it went in the right direction. However, this didn't happen in all the cases. So I'm going to show you another example where we're going to sever this chromosome arm. I remind you that SNP is inhibited in this case. So the chromosome arm now, rather than undergoing motion towards the metaphase plate, where these chromosomes are already aligned, it will be expelled exactly towards the cortex, exactly where we do not want to have these chromosomes. And this, so I'll give you some numbers here. This is what happens in these experiments. So SNP, in most cases, actually is able to undergo directed motion towards the plate, but there's a significant fraction of cells that actually move the chromosomes towards the cortex or have no movement at all. And this motion was totally dependent on chromokinesins, because on top of it, we can basically inhibit or deplete chromokinesins from these cells. And in this case, when we do the surgery, basically chromosomes do not undergo any movement. So this shows you, this simple experiment shows you that SNP is dominant over chromokinesins. Because if you inhibit SNP, you basically lock these chromosomes for several hours at the poles. But if you now engage chromokinesin, or you let the system depend exclusively on chromokinesins that work on chromosome arms, they will be able to move the chromosomes, but they do it in an inaccurate fashion. So sometimes they will move the chromosomes towards the cortex, which is the wrong direction. So this begs the question, so how does SNP guide polar chromosomes exclusive towards the equator? So just to summarize, diamine is the -N directed motor that will bring these peripheral chromosomes near the poles. So once at the pole, SNP will need to know where to go. So how does these motors sitting on the kinetochore actually distinguish these milieu of microtubules, this environment of microtubules, where they know exactly where to go towards the equator. How do they know where to go? In order to explain you this and get into the hard core of the whole experimental work, I need to introduce you this notion of the Tubulin Code. So the Tubulin Code, you probably have heard, most people have heard of the Histone Code, which basically relies on post-translational modifications of histones, and these regulate processes like transcription and gene expression. The Tubulin Code on its own comprises the combination of both tubulin isotypes encoded by different genes, and there's many of them in mammalian cells, with tubulin post-translational modifications. And I'm here summarizing some of them. I will highlight acetylation here, which happens in the microtubule lumen, but also these branch modifications, these C-terminal tails outside the globular domains of both alpha and beta tubulin. This corresponds to polyglutamylation. And most important for this purpose, I want to highlight one particular modification, which is known as detyrosination. So most alpha tubulin isotypes encode this last tyrosine residue over here. So all the gene products normally have this tyrosine at the end of the C-terminal tail of tubulin, of alpha tubulin. But there's actually catalytic machinery that uses energy to remove, cells actually bother removing this last tyrosine from some microtubules, giving rise to detyrosinated form of tubulin. It's a bit of counterintuitive that in terms of post-translational modification, you're actually not gaining any additional modification. You're actually losing something that was genetically encoded, and this is known as detyrosination. How does this detyrosination-retyrosination cycle takes place? So basically, tubulin in its soluble form is normally encoded with this tyrosine residue on the C-terminal tail of alpha tubulin. And this is able to be incorporated into the polymer of a microtubule that becomes tyrosinated. But then there's a complex that has recently been identified, this tubulin carboxypeptidase called vasohibins, bound to this regulatory peptide, SVBP, that work to detyrosinate this last residue out of the polymer. So they seem to have a preference, although it doesn't seem to be exclusive, but have a preference for these tyrosinated molecules that are incorporated into the polymer. And they're not the only players, but these are the only ones we know, because there's still some detyrosination going on, despite the fact that we could knock out completely these vasohibin genes. There's two in the case of humans. So you have, through these catalytic action of vasohibins, you have detyrosinated microtubules, and then these detyrosinated tubulin could be when the microtubule depolymerizes and it's in equilibrium with the soluble tubulin. So basically, this detyrosinated tubulin can now be recycled again by another protein called tubulin tyrosine ligase, or TTL. So this closes this detyrosination-retyrosination cycle and introduces the key players in the process. What we also knew, and back to the problem that we had before, that not all microtubules in the mitotic spindle were identical. There's actually quite significant diversity in the spindle, namely in this case, and we know this since 1986, that only these microtubules pointing towards the equator are detyrosinated. And these normally correspond, so here I'm showing here a more colorful picture of the process, and the detyrosinated tubulin is shown in green, whereas tyrosinated tubulin is shown in white. So these white microtubules are the astral microtubules, and you can see, because they're very dynamic normally, they do not accumulate detyrosinated tubulin. Only these microtubules pointing towards the equator that are starting to interact with the kinetochores here in magenta become detyrosinated, because they are about 10 times more stable than all the other microtubules in the spindle. So this provides an interesting idea, or an interesting hypothesis, that these motors sitting here on the kinetochores on the chromosomes that need to be aligned to the equator could use some spatial cues provided by deteriorated microtubules on the stable population of microtubules that actually point them the way to go towards the equator. So this was exactly our hypothesis, do deteriorated microtubules guide chromosomes during mitosis? And these are what people used to think about the spindle, that despite we knew that there's several microtubule subpopulations, we always considered them equivalent in the models of mitosis. But we introduced, the ingredient that we introduced in this model was we introduced microtubule diversity. So if you now make microtubules in the spindle diverse, where after microtubules, for example, they are tyrosinated, and these microtubules pointing towards the equator will be deterosinated, this would explain how SNP at the poles would be able to move chromosomes exclusively towards the equator. And we could add some predictions. So if we inhibit the carboxypeptidase activity necessary to deterosinate microtubules, so you render all your microtubules tyrosinated, basically this would phenocopy the inhibition of the motor, because the motor, once the chromosomes get stuck at the poles, they will not be able to move towards the equator because they miss the spatial cue that shows them where to go. On the other hand, if we now render the spindle essentially highly deterosinated, and we can do that by depleting the TTL protein that recycles tyrosinated tubulin, so all your microtubules will be deterosinated. So the chromosomes, because the motor that usually guides them uses these deterosinated tracks, the motor could now, in principle, move the chromosomes in random directions. Sure enough, they will bring some towards the equator, but many of them will be expelled towards the wrong directions, pretty much as chromokinesins did when we let the system rely on them exclusively. And so this is just to show you that we can actually efficiently deplete TTL by multiple means, and I'm showing here RNAi experiments, but we can do also CRISPR knockouts of the protein. The cells are actually viable, and these corresponds to an increase in deterosinated tubulin as expected, okay, with no much change in the tyrosinated level of tubulin, which composes most of the tubulin in the cell. We could also inhibit tubulin carboxypeptidase activity with this drug called partenolide, which basically blocks sits right here on the catalytic triad of this carboxypeptidase, preventing its protease activity and therefore preventing tubulin deterosination activity. Just to show you what we see in cells, so pretty much these movies are running in parallel. And you can see here that the partenolide, when we inhibit the carboxypeptidase, basically phenocopies the inhibition of the motor. So we end up in a situation where most chromosomes are able to align, but some chromosomes get stuck at the poles because they basically are unable to find the spatial cue that guides them towards the equator. So they phenocopy the inhibition of the motor. On the other hand, the depletion of TTL also in this case compromises congression a bit. And you can see that these chromosomes, they are no longer clustered at the poles as you see here in the SNP-inhibited cell or the partenolide, but they get scattered around the poles, being expelled towards the cortex. You can have a close-up look into these poles, and you can basically confirm here that the inhibition of the motor or inhibition of tubulin deterosination phenocopies the clustering of these chromosomes that get stuck at the poles and are unable to go to anywhere, in fact. But TTL depletion instead does not cluster the chromosomes, and they now are able to migrate in all directions, including towards the cortex. And these random motion can be is essentially SNP dependent because you can significantly rescue it by co-depleting or co-inhibiting, in this case, the SNP motor. So basically, this tells you that either inhibiting the motor or the modification that the motor uses on the tracks that takes the motor, in this case the chromosome as well, towards the equator are informative. They provide cues, spatial cues that orient the motor towards the right direction. So how do we test this in a simplified manner? So how do we test these ideas that the motor indeed is reading these cues? So we use purified detyrogenated tubulin or tyrogenated tubulin that we extract from human HeLa cells, and we did this in collaboration with Magda Magira and Carsten Janke at the Institut Curie in Paris, and purified the SEMP motor domain and tagged it with GFP. So we can see this SEMP molecule in green, and you can see here the different types of microtubules. So the detyrogenated microtubules will show up in pink or magenta, and the tyrogenated enriched microtubules will be shown here in red. And the green dots that you see here are the motor single molecules of these SEMP motors walking along these microtubules. And we're going to see that they are able to ride these microtubules towards the plus ends. But if you have a good eye and look carefully, the velocities on the detyrogenated microtubules, the pink ones, are faster. And this is calculated here. So this motor is actually more efficient motor on detyrogenated microtubules, and they're able to run significantly increased lengths if the microtubules are detyrogenated, or at least enriched in detyrogenated tubulin, as compared to microtubules enriched for tyrogenated tubulin. We could then apply force on these motors by coupling these motors to beads. So these beads can be trapped on an optical trap, an infrared laser basically, and positioned along the microtubule that has a minus end and a plus end. This is shown here. And as the motor tries to move across the microtubule, it will exert force. So basically we can measure how efficient is these beads that are trapped by the laser can try to escape the force that is being undertaken by the optical trap. And the typical experiment looks like this. I'm going to show you here the microtubule shown in red. There's a bead on top of it. There's a green signal coming out of GFP coating the bead. And you will see that so the bead will now show up only, and the bead will start wobbling and will basically trying to escape the optical trap. And we can measure these excursions, and these are shown here in these two graphs. And this is a tyrogenated tubulin, microtubule, then you can have these spikes of activity. But if you do that on detyrogenated microtubule, basically the spikes are much higher, meaning that you need to experience much higher force to counteract the loss of the bead, and the duration of the spikes is also significantly higher. So, in other words, these beads are able to detach out of these microtubules easier if they are on tyrogenated microtubules. So you need basically need much higher force to have these beads detaching out of these microtubules, as opposed if they are traveling on tyrogenated microtubules, meaning that they are more efficient motors transporting these beads along these microtubules, along, in this case, detyrogenated microtubules. So in summary, so this slide shows that we can reconstitute SEMP-mediated motility in vitro and compare its efficiency along tyrogenated enriched or detyrogenated enriched microtubules, and we show here that SEMP is a much efficient motor on traveling along detyrogenated microtubules. So this gave us a hint and a direct point to look at this in live cells again. And here I'm showing a cell that is expressing microtubules in red and SEMP and in green with GFP. And SNP in interface cells also decorates microtubules, and it looks like this. So if we basically treat these cells with DMSO, which is a solvent that we use for these experiments, so you can see that the SNP holds on pretty tight on microtubules over time. But if we now do the same experiment, inhibiting tubulin carboxypeptidase activity, you can see here that microtubules again are in red, and here SNP is in green. See what happens. Just in a matter of a few minutes, the microtubules remain, but SNP completely detaches out of these microtubules. So by changing the detyrogenated state of these microtubules in cells, you can change the ability of SNP to, in this case, accumulate on microtubules, further supporting our in vitro reconstitution data. So altogether, we proposed a model in which tubulin detyrogenation works like a navigation system for chromosomes. So if you have a system that relies on these modifications and you inhibit detyrogenation, chromosomes will be locked at the poles. But if we now make all the microtubules detyrogenated, the motor that reads these cues basically will go in all directions. So during normal division, we have a mix of microtubules. We have microtubule diversity. And these different tracks can be used to regulate different motors to allow them to find their way towards the equator. So how do we get into this idea of mitotic error codes? So this was our way into the Tubulin codes and their roles in mitosis. And now I'm going to show you the latest part of our work that was just published a few weeks ago that looks at the role of detyrogenation in other aspects of mitosis. And indeed, so just to point out that this idea of Tubulin code and navigation system might actually be a real idea. It was later confirmed that the other opposing motor, dynin-dynactin, actually has a preferential movement along tyrogenated microtubules. So the two motors that sit at the kinetochore and pull on different directions basically have distinct preferences for these microtubules. Microtubule modifications. Dynin-dynactin prefers tyrogenated microtubules, whereas SNP prefers detyrogenated microtubules. And this is how we think that the microtubules use these modifications to regulate the activity of different motors, in this case, sitting on the same structure on the chromosome, the kinetochore. So what are the aspects of mitosis are regulated by the Tubulin code? And basically, we know that there's not just one kinesin-like protein in mitosis, but there's about 12 kinesin-like proteins that have been described to play roles in mitosis. So it's pretty reasonable to hypothesize that other aspects of mitosis might be regulated by the Tubulin code by affecting these other motors involving different mitotic processes. And one motor in particular that I want to bring to your attention is actually not a classic motor. So it's a kinesin 13 protein that, rather than moving cargo along microtubules, uses the energy derived from ATP hydrolysis to disassemble, to promote the disassemble or depolymerize a microtubule. And this was shown several years ago by the group of Annie and Rieu, and more recently by the group of Ronville, that kinesin 13s are negatively regulated by detyrogenation. So they actually are much better microtubule depolymerizing motors if they have to work on tyrogenated tubulin rather than detyrogenated tubulin, as opposed to SNP, as I explained before. So this gives us a suggestion that a very important aspect of mitosis concerning error correction might be regulated by the Tubulin code, because one of these kinesin 13s, NCAC, is actually one of its favorite locations. It's on the centromere. And being at the centromere, you can actually use these capacity to depolymerize microtubules to correct erroneous attachments. So this activity would be there to prevent these kind of incorrect attachments in which a single kinetochore is attached to microtubules from both poles, and therefore these wrong attachments should be destabilized, and it's believed to be undertaken by the action of these MCAC proteins sitting on the centromeres. And therefore, when all the attachments are corrected, you can have successful segregation. But if you have MCAC activity compromised, these erroneous attachments go unsupervised and undetected, and they cannot be corrected. And this leads to several types of chromosome desegregation events, we think by compromising MCAC activity. So we hypothesize that these processes of error correction involving MCAC could be regulated by tubulin deterioration. And to address this, Luisa Ferreira in our lab started investigating the accumulation of deteriorated tubulin on different types of kinetochore microtubule attachments. And to do that, she converted the spindles in multiple configurations. Here's what we call a monopolar spindle configuration, which we can trigger by inhibiting kinesin 5 with this small molecule inhibitor called Monastery. And this gives rise to these incorrect types of attachments called sympilic attachments, in which both kinetochores are attached to a single pole, or incomplete or immature attachments in which a single kinetochore is attached to a microtubule. So it's called monotelic attachments. And here on the gray panels, you have the accumulation of deteriorated tubulin on these microtubules. And you can see it's pretty faint on these configurations. But if you now compare this signal with a signal that we normally obtain when the microtubules are, when the spindle is bipolar and the chromosomes are bioriented, you can see that deteriorated tubulin also accumulates precisely on the microtubules pointing towards the equator, as I showed before. And this is even exacerbated if we now hyperstabilize these microtubules with this drug called Taxol. So I give you some numbers. So indeed, a chromosome that is bioriented has a much higher deteriorated tubulin level, very close to the kinetochore. So we're measuring here in the very close vicinity of the kinetochore, within 1 micron distance along the kinetochore microtubules from the kinetochore, as compared in this case to a condition in which the attachments are not fully matured or are incorrect. And this is exacerbated, as I told you, by treatment of the cells with these microtubule stabilizing drugs. So we have here, in a quantitative manner, an indication that different types of attachment have different types of, or accumulate different levels of deteriorated tubulin, with more stable attachments on a bipolar configuration being enriched for deteriorated tubulin. So then we asked what are the consequences of having microtubules deteriorated? And to do that, we basically deplete TTL from the cells. And if you see carefully here, you can see that if we deplete TTL from the cells, all your microtubules suddenly become deteriorated, including these tiny little astral microtubules that are normally not deteriorated. And then, so we basically constitutively increased tubulin deterioration in the cell and asked, what is the consequence for chromosome segregation? And here I'm showing 2 movies, a control cell in which you have the green dots revealing the chromosome position of the kinetochore in this case, and here is what happens in TTL depleted cells. So the chromosomes have a hard time congressing, but they eventually manage to align. But I hope you notice that some of these green dots actually lag behind. So I'm going to play this again, and hope you appreciate that right here you have these tiny little dots of chromosomes that lag behind. And this is very frequent in these TTL depleted cells. So if you compare here in these pool from 3 different independent experiments. It seems that if you render microtubules constitutively deteriorated, that causes problems in either error correction, or you actually make the cells more prone to make errors. We actually investigated these deeper by super resolution microscopy. This is a modified version of STEV microscopy, which we called coherent hybrid STEV, a method that was developed by Antonio Pereira in our lab, where we can very easily track microtubules at very high resolution and investigate and look how these chromosomes that are lagging behind look like. And in fact, you can see that the kinetochore region here in red is actually stretched, and you have microtubules ending from both sides, indicating that this incorrect conformation called merothelic attachment is actually taking place and explaining why you have so many chromosomes lagging behind when we perturb PTL activity, or when we make microtubules constitutively deteriorated. Just forgot to mention, so we did one experiment in which it's called the Monastery washout experiment. So we treat cells with this drug Monastery. The chromosomes become mono-oriented because these spindles are monopolar, and we can now release these cells from Monastery, allowing the cells to become bi-oriented. And if you see these numbers, so if you compare untreated cells with Monastery-treated cells, you can see that both control and TTL-depleted cells increase, and they increase in the same proportion, suggesting that you're not increasing the rate of error formation by depleting TTL. So this difference here is either being caused by a problem or a compromised error correction process. And here, to substantiate these experiments, we actually measured whether centrosome separation at the beginning of mitosis is normal in TTL-depleted cells, because if you abolish centrosome separation or you delay centrosome separation early in mitosis, you make cells prone to establish erroneous attachments. And so it's a clearly error formation pathway that becomes activated if you don't separate centrosomes. And then if you can see, the centrosome separation is absolutely normal with or without TTL. So we don't think that indeed increased deterioration is contributing to erroneous attachments because they form more often. It's rather the opposite. The correction becomes more compromised. And I need to introduce you to the models of how people perceive error correction takes place. And there's basically 2 main models with a clear dominant one, which is highlighted now in the screen. So basically, error correction takes place by promoting kinetochore microtubule turnover. And there's a sort of sweet spot in which kinetochore microtubule half-life is just right to basically allow the satisfaction of the spindle assembly checkpoint, which monitors the attachments of microtubules to kinetochore, but on the other hand, prevents the hyperstabilization of the attachments, which could lead to chromosomal instability that I alluded to in the beginning of the talk. So this would be taking place on all kinetochores in a non-discriminatory manner, so that the eventual mistakes will get corrected after some time, and chromosome segregation could take place normally. There's also additional views that are perceived as discriminatory. For example, this protein Aurora B and also MCAC that I told you about, so Aurora B is a central player in error correction in mitosis, as it is MCAC. In fact, Aurora B is able to regulate MCAC activities through phosphorylation because it's a protein kinase. So you can actually found these 2 proteins, MCAC and Aurora B here, enriched on erroneous attachments. So it does suggest that the cell is able to sense in some way where erroneous attachments are and recruit the factors to these places or enrich in these places to allow for efficient error correction. So if our results with detailination were causing errors by changing microtubule stability, so in this case, the non-discriminatory model that I referred to, we would expect to see changes in kinetochore microtubule half-life after depleting TTL. So we do that by photoactivating GFP tubulin on a cell that stably expresses N-cherry tubulin. So you can see nicely the spindle here, and then we basically photoactivate a stripe of GFP alpha tubulin and measure the dissipation of fluorescence over time. And that gives you a quantitative view of the turnover or half-life of the kinetochore microtubules in these cells, because we can extract this out of these decay curves, taking into account these very slow recovery components here shown in these graphs. And you can see that if we do that, there's basically not much changes between controls and SI or TTL depleted cells, both in prometaphase or metaphase. So this basically tells you, gives you an idea of the estimated number of microtubules in both conditions. But most important, if you look at kinetochore half-life, as expected in control cells, the stability of microtubules increase as cells transit from prometaphase, this early mitotic condition, to metaphase, when they become bioriented, where the chromosomes become bioriented. But there's no difference, no measurable difference when we deplete TTL out of these cells. So we're having more errors without globally changing kinetochore microtubule half-life. And what is interesting is that indeed, if we completely deplete MCAC from the cell, you do see an increase in kinetochore microtubule half-life, the kinetochore microtubule stability, when cells are devoid of MCAC, specifically during metaphase. So these two results are a bit difficult to integrate because, okay, MCAC is involved in error correction, and as expected, it does have a role in kinetochore microtubule half-life when we deplete its entire pool, but TTL doesn't seem to act through these pathways, because it doesn't change kinetochore microtubule half-life. Another aspect that is curious is that indeed TTL depleted cells accumulate deteriorated tubulin precisely on kinetochore microtubules here illustrated in this panel. But MCAC depletion accumulates deteriorated tubulin essentially on astral microtubules. So the overall depletion of the total pool of MCAC in the cell preferentially affects the stability of astral microtubules as opposed to, in this case, the depletion of SITTL, and the pattern of deterioration is completely different between these two scenarios. So it's not going to be that the TTL and the increase in microtubule deterioration is causing an impact on overall MCAC activity. So there's still something to understand between the relationship between tubulin deterioration and MCAC. But what is curious is that if we overexpress, if we give the cells more copies of MCAC or increase or enhance its activity with a small molecule agonist UMK57, we're able to rescue the increase in the lagging chromosomes or the erroneous attachments in this case in cells depleted of TTL. So you should see, look at this bar here where we deplete TTL, we basically rescue the percentage of errors by overexpressing GFP MCAC or enhancing its activity. So somehow there's some link between increased deterioration and MCAC activity that we do not fully understand. So to dissect this, we basically used different MCAC mutants and asked about their efficiency in rescuing the TTL depleted cells. So I'm showing here the full-length MCAC. So she has his motor domain here, and the kinetochore binding domain is here on the N terminus. And we have basically a dead version of this depolymerase, so it lacks depolymerase activity. It's called this MCAC high peer motor, and also this delta N construct, which prevents MCAC to go to the kinetochore. And two other constructs here basically constitutively target MCAC exclusively to the centromere. So you can follow this localization here. You can see nicely localization of MCAC exclusively on the centromeres here. And we also have a dead version of this construct that we constitutively target to the centromeres by fusing with this constitutive protein SENB. So we use these different targeting, these different mutant constructs to ask about their efficiency in rescuing the increase in chromosome missegregation due to constitutive increase in microtubule deterioration. So as expected, so the full-length GFP MCAC rescues to some extent the errors, the anaphase errors observed after PTL depletion. But if you, and this is dependent on the depolymerizing activity. So if you have a catalytic dead version, there's basically no effect on, no rescue effect due to increased deterioration. And if you lack the capacity to go to the centromeres or kinetochores, you also are unable to rescue these mistakes. So you need both depolymerizing activity and kinetochore centromere localization to allow the rescue of error correction observed in TTL depleted cells. And most important, this seems to be sufficient also for this rescue. So if you target these MCAC to the centromeres, you'll efficiently rescue to a certain extent the errors observed in TTL depleted cells. And again, this is absolutely dependent on its catalytic activity. So in short, MCAC depolymerizing activity at centromeres is required and sufficient to significantly rescue error correction due to high microtubule deterioration after TTL depletion. And consistent with this idea, the effect that we see when we overexpress MCAC full length over spindle length or astromicrotubule length is quite notorious. So if you globally change the activity of this protein, you do affect spindle length and astromicrotubule length because you affect globally microtubule dynamics. But this construct here, the GCPBM, which was able to rescue the errors observed in TTL depleted cells, has no effect on overall spindle length or astromicrotubule length, again suggesting that the way that this construct is rescuing or reading the deteriorated state of microtubules is very localized, particularly the centromere. It doesn't affect or it doesn't involve other pools of MCAC present in other parts of the spindle, like the microtubule plasens or astromicrotubules. And in fact, this kinetochore microtubule, we can do basically the same experiment that as we did before, measuring the kinetochore microtubule half-life in cells that constitutively express the GCPBM construct, and we find no effect on, no change in kinetochore microtubule stability in metaphase compared to control cells. So basically, the microtubules adopt a similar kinetochore microtubule half-life between prometaphase and metaphase when we express this construct, and this is no different from the stability that we normally observe in metaphase. So we don't think that deterioration is globally impairing MCAC function, but it's specifically impairing a fraction or a pool of MCAC at the centromeres. So we put this into a model, and we like to refer this model to the Mitotic error code, which is the title of this presentation. And basically, to summarize this last part of the work, we envision that microtubules and the types of attachments that occur in the cells are quite different. And early on, when the attachments are immature or incorrect, microtubules will essentially be tyrosinated. And tyrosinated tubulin will promote or will favor microtubule depolymerase activity by MCAC at centromeres, contributing to the destabilization of these attachments. So it's part of this feedback loop that goes on at the centromeres. So these will contribute to the detachment or depolymerization of these microtubules, and eventually the cell will reach the desired configuration, the bioriented or amphitelic conformation. And as they become bioriented, as we showed, microtubule detyrosynation accumulates in the kinetochore vicinity, and this works as a negative feedback. So it basically prevents the ongoing or continuing activity of MCAC at centromeres, because detyrosynated tubulin inhibits its activity, and this would further stabilize the attachments, favoring this configuration that is correct. So you have tyrosynated tubulin on incorrect, detyrosynated tubulin on correct. And moreover, these mechanisms would explain how the cell avoids further merothelic attachments and avoids the incursion of new microtubules that will try to make an error. So if you have a configuration of bioriented microtubules, bioriented kinetochores with high detyrosynated tubulin, and the new microtubule that is more dynamic and therefore accumulates tyrosynated tubulin, the cell will be able to selectively destabilize these incorrect attachments and maintain the correct ones, allowing for the discrimination of the types of attachments favoring these configurations right here. So when this is constitutively high, so the detyrosynated state is constitutively high, so the cells enter mitosis with high detyrosynated level, the process of error correction is not allowed to occur at high efficiency. So the attachments that are incorrect will not be solved, and they will perdure in the cell, eventually contributing to chromosomal instability. And this work was recently published in JCB, so if you're curious, you're more than welcome to visit this paper, and I'll be happy here to also follow up with some discussion. So with this, I'd like to thank the people that did the work, and particularly Luisa Freire. And as we speak, Luisa Freire just graduated, defended her thesis just a couple of hours ago. So congratulations to her and for completing this work. And also, I'd like to thank Marin Barisic, a former postdoc in the lab, that initiated the work with tubulin modifications in our lab, and now has his own lab in Copenhagen. Several collaborators for help in study and for reagents, people from my lab, these agencies for funding, and you for your attention, and I'll be happy to take questions. So thank you very much.
I'd just like to thank Elder for his fantastic presentation. That was certainly a very comprehensive series of experiments leading to his model, the error code model. There was a fantastic combination, very interesting combination of obviously very high resolution microscopy backed up by the molecular biology with using the various inhibitors and RNAis. So very, very nice series of experiments. Thank you, Elder. So I'd like to now move on to some questions that have come in. So the first question I have is, I would like to ask if the beads would not have any effect or hindrance to the migrating microtubules. Do the beads have negligible mass and would not affect the displacement? I think this refers to earlier in your presentation, Elder.
Okay, thank you. Well, that's a very good question, but indeed, the beads are exactly the same in both conditions. So the mass is not going to be negligible. So these are polystyrene beads. They are both coated with the motor, and the difference in this experiment, the variable that is changed, is essentially the enrichment in detyrosynated tubulin or tyrosynated tubulin. So we have microtubules that are enriched for one modification or the other. The beads are the same, and they are both coated with SMP motors fused with GFP. And we just measure the displacement of the bead and out of the optical trap. So it wouldn't really matter in this case because the beads are stereotypical, so they're exactly the same in both experimental conditions. So what changes are the microtubules that are gliding on, and these can be measured using the optical trap that are calibrated. I hope this answers the question.
So the next question, are there any studies about the inhibition of microtubule detyronization in cancer cells? And does the microtubule detyronization inhibition, is it a good potential to target for cancer cells?
That's a very interesting question. Indeed, it has been shown that the detyrosination is a frequent occurrence in some types of cancer, and it is also used for prognostic reasons. And people, tumors with high tubulin detyrosination actually have a poor prognosis. And it's also known that suppression of this key enzyme, tubulin tyrosine ligase, occurs during tumor growth. So it seems that for a tumor to develop and grow, you need to suppress this tubulin tyrosine ligase activity, therefore leading to increase in tubulin detyrosination. And there's some inhibitors of tubulin detyrosination that are actually used already in the clinics. So there's the one that I mentioned, partenolide. It's a natural product that is used or has been used in some cancer studies. It's not really already used in the clinics, but it's preclinical studies, and they seem to have a good potential effect, especially in combination with other drugs used in chemotherapy that prevent tumor growth. So it might be a good strategy, since you need detyrosination to take place for a tumor to develop. It might be a good strategy indeed to suppress tubulin detyrosination. There's now more specific and more powerful inhibitors of these tubulin carboxypeptidase that take place in undertake tubulin detyrosination. So it's a very promising avenue that is still pretty much underdeveloped and deserves higher attention in the future.
So thank you, Helder. For next question, what determines the accumulation of a specific tubulin post-translation modification on a given population of microtubules?
That's a wonderful question. And indeed, it's probably a combination of factors. One key factor is microtubule stability. If your microtubules are short-lived, they will not have time to accumulate specific tubulin post-translational modifications. So you need to have a certain stability, so the microtubule has to be long-lived so that you start accumulating some modifications, in particular the one I mentioned today, detyrosination, but this is also the case for others. The other key element is, where are the enzymes located? And this is still ill explored and deserves further attention, because only recently we knew which enzymes actually account for these specific post-translational modifications. So it's a relatively new field. The enzymes became known only for a few years, and determining where they localize and what determines the localization of the enzymes on particular microtubule subpopulations would also help explaining why only a given population of microtubules accumulates a particular modification. But I would go with the first explanation that microtubule stability is certainly a key factor that determines that the microtubule will be post-translationally modified.
Okay, and there's just one final question before we wrap up, and that is, how do tubulin post-translation modifications combine to control function? That's
another excellent question. We are only starting to understand how individual post-translational modifications regulate particular functions, how the real concept of the Tubulin Code, which relies on the combination of multiple post-translational modifications together with tubulin isotypes, how this whole combination of modifications translates into function. It's one of the most important open questions in the field, and we don't have an answer yet. But now with our understanding of the mechanisms and the enzymes involved, we could envision that we could combine individual or multiple perturbations of multiple enzymes involved in different post-translational modifications, so that we can understand the power of the modifications and also take advantage of the super resolution techniques to map how the different post-translational modifications add to one another and combine on a particular microtubule to eventually regulate function. So it's going to be an open question for some time, and we hope to participate in obtaining some of these answers. Okay, so well, thank you very much for attending this first invited speaker webinar on this event. So very much appreciate you joining us, and thank you again to Elder Maiato for his presentation. And we hope that you get to enjoy the rest of this virtual event over the next couple of days. Thank you very much. Thank you.